Hi, welcome back to discussing taxonomic procedures and conservation and how important taxonomy is to conservation. We don't often think about how important taxonomy is because, usually you think of just it's the process of putting a name on an organism. And okay, so we know that organism is called X. And so what do we do with that? Okay, we can study X, we can do experiments with X, but it's still just X. But how important is that? There's different ways and different means in biology and science that we really have to know how to communicate the important aspects of these species. And so, so some of the reasons why we have taxonomy, why it's important in conservation and so forth, is because the species or the variety or the subspecies, that's the unit of conservation that we're interested in. And so we have to understand what that unit is to be able to protect it properly. We need to know what we're working with. It helps us to better understand the biology of the organisms that we're trying to conserve. For example, if we're working with a legume, something in the pea family, typically those have nodules associated with the roots that will fix nitrogen. So they're going to be very keen on growing in places that might have poorer soil. Where something like a daisy or something in that family, the Asteraceae family, it doesn't have that, and so it wouldn't do so well on those kind of soils. And so being able to understand that can be very important in where we're going to plant things and try to grow them in the future. Taxonomy helps us to understand how rich and biodiverse a particular region is. If we know that all the different species in a particular region maybe there's 100 species there, or there were 100 species there 50 years ago. But now we go back and look and there's only 30 species there. Still the dominants are there, but all the other species are gone. And so we know that there's been something going on there that's not good, and we want to be able to correct that. So the biodiversity or taxonomic richness is important. Much data is often lacking for a species, and so it helps us to target research projects to help us get more information about those. One example is breeding studies. We don't know how a lot of species reproduce. We know how a few of them do. Bees come and pollinate the flowers and so forth and so on. But there's a lot of variation in how breeding takes place for different species, and so a lot of times we need to figure these things out. But one of the most important things that taxonomy can do for conservation is this. Bad taxonomy can kill. This is the cover of a journal in the journal Nature, which is one of the most prestigious international journals in science. There's Science, the journal Science, and the journal Nature, which are the two go-to type of magazines for very important types of science, and primarily medical research and things of that nature. But in 1990, they had this issue called Bad Taxonomy Can Kill. And what this is implying is that the taxonomists aren't going out killing plants, or lack of taxonomists is resulting in killing plants or animals. What it means is if you have a bad taxonomic representation for your species, you might let something go extinct that you didn't expect to, and then you only find out later that it was a distinctive species, and therefore it did not get the protection that it really deserved, and ultimately it was able to go to extinction. And so that's one of the things that we really want to take keep a close eye on, is that bad taxonomy can kill lots of species, and it happens over and over again. In this particular case, this was the tuatara, which is a reptile that occurs down in New Zealand, and there was populations of this on little islets that were found off the coast, and they thought that it was all just one big species, and then on closer examination they realized that it was actually two different species, and so one of the ones that was not being protected was then able to get the protection it needed, so it did not go extinct under their watch. In this example, I wanted to talk about some examples in the Hawaiian flora. In particular, the genus Hesperomannia. It's in the composite family, the Asteraceae. So every flower, as you can see here, is actually composed of numerous little flowers to make up this one large inflorescence that's present here. Hesperomannia is a genus that occurs here in Hawaii. There are 3 species that were previously considered. And these were the only 3 species known in the world. 1 species was endemic to Kauai. 1 species was on Oahu and Maui. So arbuscula was on West Maui. And the other species in the Waianae Mountains and West Maui. The other species, arborescens, was found on the Ko'ola Mountains and Lanai and Molokai. So 3 species that were known at that particular time. From a biogeographic perspective, how they believe that this might have taken place is that the first colonist came to Kauai. That's where Hesperomannia lidgatii occurs at, is up in these mountains here. And then from Kauai, after Oahu came out of the ocean, it colonized Kauai, probably over to the Waianae Mountains, which are a little bit older than the Ko'ola Mountains, about 1 million years older. After they became present here, it colonized the Ko'olaus. And then each of these separately colonized islands of Maui Nui. So collectively, these are the islands of Maui Nui. And so from the west, from the Waianae Mountains, it came to West Maui. And from the Ko'olau Mountains, it came to Molokai and Lanai. This genus is not represented at all on the Big Island or Kaho'olawe. So we did our first experiments with this particular group. This is a phylogeny that we developed based on all the different populations that we could find. We have an outgroup series of species that we used in this comparison. And what this outgroup does is it gives orientation to the evolution of everything within that. And so, so you can see here that from this branch here, this is the branch that leads to all the different species of Hesperomannia. So based on the position of this outgroup to species that are from Africa. And the important part here is to notice how jumbled up it is. So the Kauai species, they are all together there in this one branch, so that's good. Hesperomannia arbuscula from the Waianae Mountains is all together here in this other branch, except for this one down here in Molokai. So that's not good, having this separation of the species here. And then arborescens we have in the Ko'olau Mountains, but heck, we also have it down here on Maui too. And so this group here with both arbuscula and arborescens, and this group here with both arbuscula and arborescens, which really doesn't make any sense in terms of an evolutionary process. How could this have happened? It just doesn't imply how these relationships could have occurred naturally. So what we did was we went with the assumption that this colonization took place as we would expect. And so we realigned the different species in this particular study. We published this in Pacific Science a number of years ago. Pacific Science is a journal that's published here in Hawaii, and mostly on Pacific-related species. But here you can see with this change, we have all the Kauai species, they're together in this one branch. We have all the different collections of Maui Nui here on this one branch. And then the Oahu species are on this branch, this one group here being the species in the Waianae Mountains and all the other species here, or all the other populations here representing the Ko'olau Mountains. We looked at this, so this is how we use the sequence analysis, looking at different genes that are present in one individual from each of the populations that were studied here. We didn't just collect one individual from each population, but we only sequenced one. But then we did use a population type of analysis to look at this a little bit more fully. And that's represented here. This is using RAPIDS, random amplified polymorphic DNA. And we were able to then, with this, utilize the variation that occurred among all the different markers. We analyzed about 100 different genetic markers and the presence or absence of the bands within each of those markers to determine this relationship. And when we ran this through a program, this is a principal coordinates program, which helps to redefine the ordination of the individuals there. Every dot here does represent a different individual from those populations. And you can see that we looked at lots of individuals. So it turns out these are all the individuals from Kauai, representing Hesperomannia ligatii. These are all the individuals from Oahu, and I'll talk more about those in just a moment. And these are all the individuals from Maui Nui. And you can see with this particular ordination that the three island groups broke out from each other really clearly. And so it's very obvious that the Arbuscula from Molokai and Oahu cannot be closer related, because this is the Oahu and this is the Molokai populations over here. And the same thing for the arborescens from Molokai, I guess it's Arbuscula from Maui and the Waianae Mountains, and arborescens from the Ko'olau Mountains here, and arborescens from the Molokai region over here. So again, the islands do separate very clearly from each other. When we look just at the Oahu specimens, we saw something very interesting there. On the right, these squares are all the different populations and individuals from the Waianae Mountains. And over here, these circles represent the different individuals from the Ko'olau Mountains. And so you can see all the populations in the Ko'olau Mountains are on the left, and all the three different populations from the Waianae Mountains are on the right. Again, giving a lot more strength to the idea that on Oahu, it isn't just one species, but it is two distinctive species that are represented here. And it's also interesting how even a little bit further in the Waianaes, you can see the different populations do kind of separate out from each other a little bit more clearly there as well. So the results of this study showed that there are not just 3 species, but 4 species that can be recognized in the genus Hesperomannia. And this is important from a number of different perspectives. One, just understanding the biodiversity, the evolution that's taking place here, the biogeography of the species, which I'll get into a little bit more in just a minute, but also the protection of the species. So now we recognize arborescens on Maui Nui. This would be Molokai and West Maui. There was the species represented on Lanai, but that has been looked for extensively and not found. So it's believed to be extirpated from Lanai. On Kauai, we have the one species Ligatii, and on Oahu, we have Hesperomannia sweezii in the Ko'olau Mountains, and Hesperomannia oahuensis from the Waianae Mountains. Sweezii and oahuensis are old names. We did not rename these as new species. Back in the history of the taxonomy of this group, these populations were named for a collector named Sweezy, who actually collected some of the plants for this population here. And then oahuensis is the name that was applied to the plants in the Waianaes. And so we are really just re-recognizing these names for these individuals now. From a conservation perspective, though, it's really critical because Oahuensis, which was tied to Arbuscula, which was on Maui Nui, arborescens takes priority in terms of a name, so the name Arbuscula is no longer valid. But when these populations were shared with this other, it was not considered to be an endangered species. But now, since it's a distinct species in the Waianae Mountains, we also recognize that it's critically rare. There's these 3 populations that were studied, and those squares that were represented on this figure here, these are literally all the individuals that were known at the time of this study were included here. And you can see there's only like, what, about 35 individuals that are present. And now, since these collections were made about 15 years ago, a number of these individuals have since died. And so the size of these populations are much, much smaller than they used to be. And so because of that, we can now legitimately give very good restoration attention to the conservation of Hesperomannia oahuensis. I wanted to mention also some of the aspects of the biogeography that's taking place here. And this is really pretty cool to think about. One of the aspects is that Maui Nui used to be much larger than it is now. This is actually looking at Maui in the past, so about 1.2 million years ago, where it literally was one very large island. Since then, the sea level has risen a little bit, but mostly the islands have settled down a little bit. And so through island subsidence, they are starting, our islands are every day sinking a little bit down into the ocean, onto the ocean floor. And so you can see here the outline of, let's see, I guess this would be Maui here. This would be Kaho'olawe. This is Lanai, and this is Molokai over here. Where they would have been 1.2 million years ago. So one of the aspects of this is Hesperomannia colonized to here and here. This was probably one continuous landmass through here, as you can see here at this particular time. And so it was very likely that the connectivity between these island or these mountain masses here was probably consistent. And so you had movement of individuals throughout West Maui and Lanai and Molokai to make this just a single species, which then when the island levels or the water levels have receded, became separate and no longer are able to connect with each other anymore. About 18,000 years ago, this is what things looked like. And so again, you can see here's West Maui, here, Haleakala there, Kaho'olawe distinct at this particular time, but Lanai and Molokai still attached to Maui through this last glaciation period. And then we see it now as it is now, as it is here. In terms of the biogeography, how we could look at this now is very interesting. So we know that based on the phylogeny that colonization to Hawaii took place pretty recently. The lines between each of these different groups, the Kauai group, the Oahu group, and the Maui Nui group, was very rapid. And so there wasn't a long period of time between colonization from one to the other to the other. And at the time of colonization, about 1.5 million years ago, Maui Nui was the largest target that was present here in Hawaii. And so very likely, instead of having a colonization from someplace else to Kauai and then to Oahu and then to Maui, colonization probably occurred here first and then went back to Oahu and then back to Kauai. There's some morphological evidence that's tied that could be used to corroborate this. although nothing very definite. You know, we can't genetically say that that's absolutely how it happened. We don't have the evidence for that at this particular time, but it's worth looking into a little bit more in the future. Okay, so that's the story of Hesperomannia, going from 3 species to 4 species. And I hope you enjoyed this presentation.